Welcome to our department seminar. Today, we are happy to have Dr. Amaya Gondhalekar from Purdue University to give us a seminar about bedbugs. I would like to give a very short introduction about him. So Amaya is a research associate professor at the Center for Urban and Industrial Pest Management at Purdue University. Actually, I was in the same position more than 14 years ago. So he basically took the position I took before. So I have been known Amaya from 2008. Around that time, he was studying PhD at University of Florida. So he was originally from Mumbai, India, where he got his bachelor degree in agriculture and entomology. Then he got training in urban entomology at the University of Florida to work on cockroach toxicology and resistance management. Then after a 1-year postdoc at Purdue University, he was promoted to a research assistant professor. Now he's a research associate professor. So his research program focuses on the basic biology and management of urban pests, including cockroaches, ants, bedbugs, etc. So today he will talk about bedbug. So Amaya?
Yeah, thank you, Dr. Changlu. Thanks for inviting me to present in the seminar series at Rutgers Entomology. I just was talking to Dr. Hamilton before a few minutes ago, and I learned that this is kind of also a seminar series or a seminar course for undergraduates and graduates, which is a pretty good idea to kind of get students used to attending seminars and just talking with the seminar speaker. So without further delay, I would like to get started on today's presentation. I'll minimize my video, that way it's not interfering. So here is the outline of my seminar. Dr. Wang gave a brief outline of my program, but I'll go a little more in depth and talk about some of the research projects that I have been doing recently and over the years. Then I'll talk about significance of bedbugs as urban pests, giving introduction to about bedbugs to a department which has a very world-class researcher, Dr. Wang, who does research on biology and management of bedbugs, is like preaching to a choir. But for some of the new students in this group, I'll kind of briefly introduce them. Then I'll talk about some background information on bedbug heat treatments, just as a segue into the main topic, the research topic of this presentation, which is understanding thermal biology of bedbugs. In that main section, I'll talk about 3 different projects that my PhD student, who is now an assistant professor at LSU, has done. And I'll be specifically talking about the ability of bedbugs to develop physiological heat resistance, their behavioral responses to heat exposure, as well as the role of alarm pheromones in heat-mediated escape responses that they show. I'll end this talk by giving you an overall summary about the research that we have done on bedbug thermal and chemical biology. So let's start by talking about a little bit about my lab, the research that I do. So as Dr. Wang mentioned, I work on basic biology and integrated management of urban pests such as cockroaches and bedbugs. Here at Purdue Entomology, we have 3 other entomologists or 2 other entomology faculty, urban entomology faculty that also work with different urban pests. So each of us have a niche area that we work in, and my role is to work with cockroaches and bedbugs. I was trained as an insect toxicologist and physiologist working with cockroaches. So for a long time now, I have been involved in German cockroach insecticide resistance management project with different manufacturers. And more recently, I have also started some essential oil toxicology work with bedbugs. In terms of insect physiology, some of my students who have recently graduated have worked with allergen biology of cockroaches, as well as one of the students has worked on thermal biology of bedbugs, which is what I'll be talking about today. So when I do research with toxicology and physiology, my research spans both organismal and suborganismal levels, as well as I use various techniques in molecular biology, proteomics, and analytical chemistry for my research. A slightly different, a bit applied aspect of my research is urban integrated pest management. So in this particular research area, I test various chemical and non-chemical control techniques for bed bugs and cockroaches in public housing and other institutions. Although this is a very applied area, sometimes when I'm working out in the field, I do get some ideas for basic research in toxicology and physiology that I can do with bed bugs. So although these are a bit unrelated or distantly related research areas, they kind of feed off each other. In addition to doing hypothesis-driven research here at Purdue in this position, it's a research faculty position. So I also have a responsibility of liaising with the urban pest management manufacturing industry in developing new solutions for urban pest control. So in addition, so most of my research is also involved in working with the industry that never gets published. Some of it gets published, but some of it doesn't. I have a heavy involvement of graduate students, undergraduate students, as well as postdocs in my lab. So since 2013, since when I have been in this position, I have had 4 master's students who have graduated from my lab, 3 PhD students, as well as 2 postdocs that I have co-advised. I also employ a lot of undergraduate technicians in my lab to take care of the bedbug and cockroach colonies, as well as help me with various insecticide research that I do for manufacturers. Some of these students stay on as capstone researchers in my lab, so it's kind of a win-win situation. So with that brief introduction, let's start by talking about very briefly about the significance of bedbugs as urban pests. So as most of you may be aware of, bedbugs belong to a small hemipteran family called Cimicidae. It has about 100 species in it. And some of the species, such as the Cymex lectularius, as well as the tropical bedbug, the Cymex hemipterus, are obligate blood parasites on birds, animals, as well as humans. For a while, from about 1960s to until the turn of the century in the early 2000s, most of the pest management companies or pest control operators did not have experience treating for bedbugs. This is because we were using DDT, the first organic synthetic insecticide for pest control, for household pest control since the 1940s to 60s. It was very effective against bedbugs and had almost wiped out the bedbug infestations from the households in the United States. So most of the people were not even familiar with how to control this pest or identify this pest. But in developed countries in North America, as well as some countries in Europe, as well as in Australia and New Zealand, we have been seeing global resurgence of bedbugs since the turn of the century. There are many reasons why this resurgence has occurred. One of the reason is increased global trade. Other reason is increased use of used furniture or secondhand furniture. But one of the reasons that is kind of shown to be scientifically proven that has contributed to the resurgence is their insecticide resistance. In a couple of slides, I'll talk about insecticide resistance, but for now, I want you to remember that insecticide resistance is one of the important factors in the resurgence. There are issues, social and medical issues associated with bedbug infestations. If anyone has bedbugs, they are going to see it as a social stigma and they won't mix up with a lot of people. That also leads to anxiety and depression as well as sleep loss. If I'm feeding bedbugs on myself, I don't even react if I'm allowing 1 or 2 bedbugs to feed off me. I don't get any itchy welts or any other reaction on my skin. But some people, or most of the people, do get red itchy welts, which can be very itchy, and the excessive scratching can lead to secondary bacterial infections. Some of the work that was done in Dr. Changlu Wang's lab also showed that some people can have systemic reaction to bedbug bites, which requires immediate medical attention. Some of these pictures are from that paper. Luckily, we don't have any pathogens that bedbugs can transmit to humans systemically. But recently, they have been shown to produce histamine, or in their fecal matter. So histamine has been shown to be one of the contact pheromones that is found in their fecal matter. And this histamine in the fecal matter can end up in the household dust, and then it can expose the people living in those infested apartments to histamine. What the medical impacts of this bedbug produced histamine on humans is not yet known, but researchers in Kentucky and some other universities are studying those effects. So with that brief introduction, I would like to kind of switch gears to insecticide resistance. So I said that insecticide resistance was one of the major factors in their resurgence. So when bedbugs researched at the turn of the century, we had already banned the indoor use of organophosphate and carbamide class insecticides for pest control due to the Food Quality Protection Act. So the only group of chemicals that was available for their control was pyrethroids. So soon after pest management professionals started using pyrethroids for their control, it kind of, there were reports that bedbugs were highly resistant to deltamethrin, which is one of the pyrethroid insecticide. This resistance was supposed to be caused by point mutations in the sodium channel gene of bedbugs where pyrethroid is supposed to bind. But later on, people also showed that resistance to deltamethrin and other pyrethroids was caused by metabolic and cuticular resistance mechanisms, such as detoxification of insecticides, as well as physiological changes in insect cuticle, which prevented adequate intake of pesticides into the insect body. Later on, neonicotinoid resistance was reported in bedbugs. And more recently, some of my students showed that there was reduced susceptibility in some bedbug populations to one of the newer chemicals, such as chlorphenapir, which is a mitochondrial toxin and leads to uncoupling of oxidative phosphorylation or energy production in the mitochondria of bedbugs or the cells. So because of these widespread reports on insecticide resistance, soon a lot of researchers started working on what can we do to better control bedbugs, even if they are showing resistance to some pyrethroids. Some of the seminal work was done by Dr. Changlu Wang's lab while he was still here at Purdue University. And this research showed that if you are going to combine insecticides along with some non-chemical or alternative control measures, then you can get a more sustained suppression of bedbugs as compared to the insecticides alone. There are many other papers that Dr. Changlu Wang's lab has published at Rutgers. I have not shown all of them here. When I started working here at Purdue in 2013, I worked with Dr. Changlu Wang in kind of finishing a project that he had kind of going on with another faculty whose position I took over. In that particular study, in a high-rise building, we used various non-chemical techniques such as steaming, the use of mattress encasements, as well as regular monitoring along with insecticides. And that showed that we could suppress bedbugs at a much better level if we use IPM as compared to the insecticide-only approach. So I've been talking about a lot of alternative control techniques here. And some of the alternative control techniques that Dr. Changlu Wang's group and some other researchers have recommended is the use of silicates, which kind of abrade the insect cuticle and lead to water loss, the use of entomopathogens such as Beauveria bassiana, essential oil products, vacuuming and trapping is another method, as well as steam or heat treatments at high temperatures, as well as freezing treatments. So for this talk, I would like to kind of dig a little bit deeper into heat treatments. So let's talk about some background information on how heat treatments are used for bedbug control. So one of the way in which heat is used for bedbug control is steam. So any steam machine that you can use for ironing your clothes or for other household uses can also be used for killing bedbugs that are kind of not hiding deep within the furniture. As soon as you blow steam over bedbugs, and the temperature of the steam can be more than 100 degrees Celsius or at least 100 degrees Celsius, you can see the bedbugs pop immediately as soon as the steam is touching them, because at this high temperature, they cannot resist it. There are other ways in which heat is used for bedbug control. For example, it could include heating the entire house or certain rooms within the house with a dry heat that is kind of pushed in through these blowers or heaters mounted on the truck. The goal of this whole house or whole room of treatments, heat treatments, is that you are heating the entire house to a temperature of 50 degrees Celsius or higher for a period of several hours, 6 to 8 hours at the most. Uh, you can also have portable chambers in which you can put infested furniture and do the same thing, heat up all the furniture for at least 50 degrees Celsius or higher for several hours. So when heat is employed properly, it is a very effective control measure for bed bugs because it targets all life stages of bed bugs. Some of the insecticides that we use do not kill the egg stage, but if you are using heat for controlling bed bugs, and if it is properly done, then it can also get rid of the eggs, which is important to achieve complete control. From the resident perspective, it requires minimal prep time. So whenever we are doing chemical or IPM treatments, we are the PMPs, the pest management professionals, are asking the residents to kind of prepare their apartment so they can thoroughly apply insecticides. In case of heat treatments, there is a lot of prep work that the PMPs have to do, but from a resident perspective, they have to do a minimal prep work. And heat can also be effective against some of the other household pests, such as spiders. Some people also report cockroaches and ants getting killed with heat, but they are able to escape most of the time. Some of the disadvantages of using heat are that it offers no residual protection, meaning that if you have a new introduction of bedbug 1 day after heat treatments are done, just having done the heat treatments on the previous day is not going to kill any new introduction. So it doesn't provide any residual protection. It is time intensive and costly from the perspective of a pest management professional. It can take a whole day, and the residents required to pay much more for heat treatments than a conventional insecticide treatment. So at Purdue, we have an annual pest management conference, and I have been attending it since 2012. And since about that time, some of the PMPs who are attending that conference kept telling us that they have problems uh, have with heat treatments wherein they are doing everything correctly. They are raising the temperature of the entire room to greater than 50 degrees Celsius, but still they have some survivors after doing a heat treatment. That continued for 2 or 3 years. So we decided to kind of take a closer look into why bed bugs were surviving heat treatments in the field. So some of the hypotheses that we had surrounding why bed bugs are surviving heat treatments were that, are they surviving because they are developing physiological heat resistance, or are they surviving because they are behaviorally adapted to escape lethally heated areas in the house and go find cooler areas where they can hide out and survive the heat exposure? Or could there be a role of alarm pheromones in bedbug survival? So these were the three questions that turned out into a PhD dissertation topic for my student, Aaron Ashbrook. So Aaron first did his master's with me at Purdue from 2013 to 2015, working on bedbug insecticide resistance. And then I was fortunate enough to kind of lure him and ask him to stay with me for his PhD, wherein he did some of this bedbug thermal biology work. He graduated in 2020, and then after a short postdoc at NC State University, he is now a extension assistant professor in urban and peri-urban entomology at LSU. Some of the work that we did was funded by the Pest Management Foundation, as well as Aaron's assistantship was covered by the Housing and Urban Development grant that I had with other faculty for working on cockroach insecticide resistance. So these are the funding agencies that directly supported this project. So now let's start talking about the different research topics or research aspects that we dwelled into or researched thoroughly to find out why bedbugs were surviving heat treatments in the field. And the first topic is, are they surviving heat treatments because of the development of physiological heat resistance? So one of the first thing that we had to do, or Aaron had to do, to enable him to determine the resistance of bed bugs to heat in the laboratory is that he had to develop a bioassay method. So we developed a very simple bioassay method wherein bed bugs were held in a test tube along with a filter paper as harborage. This test tube was sealed with a parafilm so that the bed bugs could not escape, but the airflow was still happening. And then these test tubes were immersed in a rack, and that rack was placed in a water bath that was heated to 45 degrees Celsius or 113 degrees Fahrenheit. The temperature of 113 degrees Fahrenheit or 45 degrees Celsius happens to be the bedbug CT max, the thermal maximum of bedbugs. That's why we chose this temperature. After exposure in this water bath for anywhere between 10 to 30 minutes, we then transferred the bedbugs to a petri dish, and they were held in that petri dish for 24 hours, after which mortality was measured. So we are not kind of measuring the mortality immediately, but 24 hours after exposure. That way, some bedbugs that were stunned by heat had a chance to recover. So this is the example of data that we collected. In this graph, you can see exposure time along the x-axis and the percent mortality along the y-axis. So basically, we were collecting exposure time versus mortality data at a temperature of 45 degrees Celsius. At 10-minute exposure, we got just under 20% mortality. And as the exposure time increased, we were able to get up to 100% mortality, after which it plateaued. We then use this exposure time mortality data to conduct a probit analysis in SAS. What probit analysis allowed us to do is that it allowed us to calculate the lethal time that was required to kill 99 to 100% of bedbugs. The data that I'm showing here is for the Harlan susceptible bedbugs. So these bedbugs have been maintained in the laboratory for more than 40 years without any insecticide exposure. And we use this population as a baseline population for any work that we do in the lab. So for this Harlan population, the time required to achieve 100% mortality as calculated by probit analysis was about 23 minutes. We used the same procedure then to determine the exposure time mortality data and the LT99 values for 9 other populations that were collected from the field. Some of the populations that we had collected were resistant to insecticides. Some of them had survived heat exposure in the field, and some of them had no history of heat exposure or insecticide resistance. So this graph here shows you the lethal time values to achieve 99 to 100% mortality in different bedbug populations collected from all over the country. As I told before, 100% or 99 to 100% of the Harlan susceptible or baseline population bedbugs were killed within 23 minutes. If you look at the spread of these LT99 values, it ranged between about 23 minutes to almost 30 minutes for the bedbug populations that we screened. We did a statistical analysis, which is a very standard analysis that is done with the LT99 or probit data, wherein we compared the LT99 values statistically between these populations. Unfortunately, we did not see any statistical significance between the LT99 values of different strains. And as you can see here, the spread, the variation was also not that much. So based on this, we concluded, or this was our first evidence that showed us that bedbugs have a very limited ability to tolerate heat, no matter their previous heat exposure history or insecticide resistant status. And because of that, our first conclusion through this experiment was that they are not developing any level of physiological heat resistance. We did a bunch of other experiments as well to prove this thing, and we found the same results. So we did not find any evidence for physiological heat resistance. But I would like to also talk about one of the other experiment that is kind of mentioned in this paper. Whenever we have to select a particular strain of insect, maybe it's cockroaches, bedbugs, or any other insect for insecticide resistance, what we do is that we subject it to sublethal insecticide doses over several generations. We did the same thing for selecting for heat resistance in the laboratory, wherein we exposed the Harlan strain nymphs, not the adults, to sublethal temperatures. So the sublethal temperature and exposure time combination was 45 degrees Celsius temperature for 17 minutes. After exposing them for 17 minutes, we were getting about 20 to 25% survivorship. So on the y-axis here, it's percent survivorship, not percent mortality. So after sublethal exposure in the parental generation, we are getting about 20 to 25% survivorship. We allowed these bedbugs to breed, and in the F2 generation, we again subjected them and F3 generation to sublethal heat. We are expecting maybe a similar response, 20 to 25% survivorship, but in fact, the survivorship went up to 40 to 55% at F2 and F3 generations. So we are a bit excited at this stage, and we thought we are going to select a particular strain, the Harlan strain of bedbugs for resistance. But to our surprise, and as it happens in most of the research, beginning at generation F4 to F7, instead of seeing any increase, we are seeing decline in survivorship. And they eventually reverted back to the parental generation survivorship levels of 20 to 25%. At this F6 and F7 generations, the heat-selected bedbugs were so compromised physiologically that we couldn't continue the selection further. For example, these heat-selected bedbugs did not feed well, they did not reproduce well, their fecundity was pretty low, which has been kind of previously shown that heat-exposed bedbugs lose their symbionts such as Wolbachia, and that reduces their fecundity. And also there were developmental problems wherein they were dying during the final molt from nymphal stage to the adult stage. So this was another evidence that kind of allowed us to conclude that bedbugs are not really developing physiological heat resistance. And so we were able to rule out the cause of bedbug survivorship after heat treatments in the field being caused by physiological heat resistance. So this kind of data is all presented in the first paper that Aaron published in the journal PLOS One. So the next, our next goal was to find out whether bedbugs are surviving heat treatments because they are behaviorally adapted to escape from lethally heated areas and find cooler spots randomly to kind of survive heat exposure. So if you look at the test tube bioassay method that we use for physiological heat resistance measurement, you can see that the bedbugs were kind of trapped in this test tube. They couldn't really behaviorally respond to heat exposure. So to determine the ability of bedbugs to respond behaviorally to heat exposure, lethal and sublethal heat exposure, we developed this copper arena. I'll briefly explain how this copper arena was made. So at the base of the copper arena was made out of a copper sheet. The reason why we used copper was because it is a good it shows good thermal conductivity. That's why we chose copper as the metal of choice. At the center of this arena, there was a harbor edge, which was made out of copper tubes, and it was infested with bedbugs before starting these heat exposure experiments. Also, in this particular arena, we had filter papers and plastic cups placed at various distances from the harbor edge. So filter paper and plastic doesn't show good thermal conductivity. So as we were kind of heating this particular heat arena, using this circulating water bath from room temperature to about 55 degrees Celsius temperature, so just above the bedbug CT max, while the arena was getting heated to a temperature of 55 degrees Celsius during the 25-minute experiment, the cups, the plastic cups, because they have a lower thermal conductivity, they were not getting heated as much. The maximum temperature of these cups was around 33 to 39 degrees Celsius at the end of the experiment. So in short, these plastic cups kind of served as the cooler spots for bedbugs to escape to during the heat exposure regime. I forgot to mention about the circulating water bath. So the underside of this copper arena had copper tubing glued to it or welded to it. And the circulating water bath was connected to the tubing, which ran under the copper sheet. And this allowed us to heat the copper arena from starting from room temperature to about 55 degrees Celsius in about 25 minutes. So using this heat arena, we exposed 5 or 6 different bedbug populations to increasing heat or ramp up heat from room temperature to 55 degrees Celsius. And what we found with different populations was that right around the time when the temperature of the harborage, the copper tubes, which was where the bedbugs were resting, as soon as the temperature was reaching 45 to 49 degrees Celsius, remember 45 degrees Celsius is bedbug CT max. So right around the bedbug CT max temperature range, the bedbugs that were resting on top of this harborage tube or inside of it were kind of fleeing this harborage because it was becoming too hot for them. So what happened when they escaped this copper harborage, the sheet or the base of the copper arena was also heated to a temperatures that were similar, 45 to 49 degrees Celsius or higher. So as they were escaping this particular harborage, they were dying because the copper arena, the surface of the arena was also lethally heated. However, around 4 to 10% of them were able to successfully escape to these plastic cups and survive the heat exposure. So let me show you the temperature ranges. So as I said, we tested 6 different populations. The maximum temperature at which the bedbugs of the Harlan strain fled the harborage was 45 degrees Celsius. And it was significantly higher in this poultry house strain. They fled the harborage completely at 49 degrees Celsius. So there was a statistical difference in between the Harlan and the poultry house population at the temperature at which they fled this copper harborage. However, that difference in temperature did not translate to increased survivorship because irrespective of which population we are looking at, we had anywhere between 4% to 12% bedbug survivorship that was not significantly different between populations. And they survived by randomly finding these plastic cups as they were kind of trying to flee the heated harborage as well as the heated arena. So what this results meant to us, or what these results showed from a practical perspective, is that when you are heat treating an apartment, say suppose a sofa or other furniture, many kind of items in the household or the room that is being treated, all of them are not going to have the same thermal conductivity. And that is going to translate to differences in their heating rates, like how soon they are going to get heated to lethal temperatures. So what we think based on these results is happening is as soon as in a real world scenario, as soon as the temperature of the bedbug harborage reaches anywhere between 45 to 49 degrees Celsius, they are kind of running out of their harborage. And the outside air in a heat treatment scenario is also heated. So some of them might be dying from heat exposure through convection. But some of them, if they are able to randomly find cooler spots as they are trying to escape, if they're randomly able to find these cooler spots, they may ride out the heat exposure without dying, right? And that could be the mechanism why they are surviving. A few of them are surviving following heat exposure in the field, even if everything is done correctly. So we also looked at the heat shock protein gene expression following sublethal heat exposure. So let me first introduce you to what heat shock proteins are. So heat shock proteins act as molecular chaperones, and they help the proteins and other cellular components from degradation or aid in their repair after stress exposure. And definitely heat is one of the stressors that we used in our experiment. One important point I would like to make is that most HSP proteins are dependent on ATP, which is cellular energy, for their functionality. So they are ATP dependent enzymes. So for our experiments, we exposed 3 different populations, a laboratory population and 2 other field populations to sublethal heat in the same arena. And then after exposure of these bedbugs to sublethal heat, we extracted RNA and did qPCR to determine the expression of 12 different heat shock protein genes that are found in the bedbug genome. So here are the results of that qPCR gene expression study. So out of the 12 different genes that we tested for, 3 different HSP genes showed differential expression following sublethal heat exposure. So the exposure was from 23 degrees Celsius to 47 for 15 minutes. That stunned the bed bugs, but did not necessarily kill them. So this is the heat map of the results from the qPCR experiments for the three genes that showed differential expression. So starting at 15 minutes after exposure, we were seeing some upregulation. So in this heat map, if you see light green color, that means there is no significant upregulation of those genes or higher expression. But if you see a dark red, dark green, or red color, that means those genes are significantly upregulated. So at 15 minutes, we had some upregulation that also continued for at 2 hours. And these different columns, I think the labels went off, they represent the different populations, the Harlan, the KVS, and the poultry house population. So even at 2 hours, all the three genes were upregulated. Same story with 4 hours. Some of the genes like HSP 70.3 started getting downregulated. At 8 hours after exposure, only the putative HSP was upregulated. And by 24 hours, that expression levels of these upregulated HSPs had gone back to baseline levels. So one of the mechanism, I think, of these or HSP upregulation following heat exposure is that, let me go back one slide. So these bed bugs, which are kind of escaping to the cooler spots, I think they are getting exposed to sublethal temperatures. And what the HSP upregulation is doing is that it is helping the bed bugs to recover from the heat stress. So from a practical perspective, I said before that for HSP proteins to function properly, they are dependent on cellular energy, such as ATP. But we also have some insecticides that affect mitochondrial energy production, such as chlorphenapir, which is a pyrrole group insecticide. So if you were to use these pyrrole insecticide or phantom or chlorphenapir before and after heat treatments, you could impact the recovery of bed bugs that are surviving heat treatments by escaping to cooler spots by affecting the functionality of their HSPs. So in conclusion, regarding the behavioral adaptations of bed bugs to survive heat exposure, we think the most definitive cause of that survivorship that PMPs are seeing in the field is their escape to cooler spots as soon as their harborage temperatures are reaching that level of 45 to 49 degrees Celsius. These bed bugs that are escaping to cooler spots, the heat shock protein gene upregulation that they show, could potentially help them mitigate the adverse effect of heat exposure or heat stress. So what can PMPs do to prevent this escape of bed bugs when they are doing heat treatments? They could easily deploy traps like the climb up traps or any other traps next to the infested furniture with the anticipation of catching the bed bugs that are trying to flee the lethally heated areas. They could be using dust insecticides as well as some insecticides such as phantom, which affect cellular energy production to kind of impact the recovery of bed bugs that are surviving after heat treatments. So in the last section of this talk, I would quickly like to talk about the potential role of alarm pheromones that are produced by bed bugs in heat-mediated escape behavior. This research is not published yet, but within the next month or so, we'll be submitting it for consideration by scientific reports. So one of the things that we kept noticing all the time, and PMPs have also noticed this in the field, whenever we are doing heat exposure experiments in the lab, we are seeing that bed bugs that were dying from heat exposure or bed bugs that were sublethally exposed to heat were kind of releasing alarm pheromones. Alarm pheromones are kind of signal danger to other not conspecific bed bugs or other conspecific insects within the species to flee the area of danger, right? So we are kind of repeatedly seeing this. We had this information from PMPs as well that they release alarm pheromones as they are dying. So we want to experimentally test this by exposing bed bugs to lethal as well as sublethal heat. And we are lucky enough to have some access to a state-of-the-art mass spectrometry facility at Corteva Agri-Sciences, which is based in Indianapolis, so just 1 hour drive from Purdue. So to experimentally test if bed bugs were releasing alarm pheromones in response to lethal or sublethal heat exposure. We placed a bed bug on a heating pad or a hot plate, and that bed bug was kind of secured in place by covering it with an inverted glass funnel. The other end, the open end of this inverted glass funnel, was kind of attached to this soft ion flow tube mass spectrometer. So this tubing that came from this particular mass spectrometer was attached to the uh, top of the funnel. So what was basically happening is that this SIFT MS machine was sampling the air coming out of this funnel as the bedbugs were getting heated. This is a very state-of-the-art costly machine. It costs about maybe 400, 500,000, or maybe even more. So it was able to do the mass spectrometry in real time. Usually you have to use PME fibers to collect the air samples or collect the chemicals in the air sample and then do a GCMS later on. But with this machine, we are able to analyze the air samples in real time. As we are exposed, when we exposed bedbugs to lethal and sublethal temperatures, we are finding that it's close to their CT max time. As they were dying, they were releasing this blend of hexanal and octanol, which are the major components of the bedbug alarm pheromone. We found this in males, females, as well as field collected bedbugs. But when we were doing blank experiments, either with bedbugs that were previously dead or blank funnels, we are not getting any signal. So this most definitively showed that after lethal or sublethal heat exposure, bedbugs are releasing alarm pheromones. I have not shown you the data for sublethal exposure, but basically the trend was the same. They released alarm pheromones at about 41 degrees Celsius. So then what is the practical implication of this particular alarm pheromone released by bedbugs in response to heat exposure? So we did some behavioral experiments where we paired some control bedbugs. So alive bedbugs were paired with other recipient bedbugs, which were also alive. So, and this was kind of a drop assay. So there was already a bedbug in there. The recipient bedbug was already in this funnel. And then this kind of a releaser or emitter bedbug was dropped in there. In control experiment, the emitter bedbug was not releasing any alarm pheromones. And you can see, hopefully this will play. Yeah, I think there is some issue with. So what basically happened in this control experiment, as soon as we dropped another bedbug in here, the male bedbug that was sitting in the funnel got attracted to it because it was recently fed, and recently fed adult bedbugs or nymphs are supposed to be very attractive to other adult males. They try to mate with it. But when in the treatment experiment, when a bedbug that was releasing alarm pheromones was kind of dropped in this particular funnel assay, the recipient bedbug, the recipient conspecific, tried to run away from the bedbug that was releasing alarm pheromone. So we are able to show that alarm pheromone released by heat exposed bedbugs were able to repel some other bedbugs. However, this kind of range, the active range for this repellency was only 5 to 10 centimeters. So it's a small active space for bedbug alarm pheromones. This escape response from alarm pheromones was also kind of proved to be shown by synthetic blend of alarm pheromones, so hexanal and octanol, that was applied to this funnel assay. So in conclusion, for the alarm pheromone experiments, we are able to show that lethally and sublethally exposed bedbugs release detectable levels of alarm pheromones. However, the active space of these alarm pheromones is very small, only up to 10 centimeters. And from a practical perspective, I think the short range of alarm pheromones is kind of significant, because if you know how bedbugs aggregate in the field, is that there are very small aggregations, number of aggregations that are present. And the size of those aggregations could be very well less than 10 centimeters. So to signal danger to conspecific bedbugs, that active space of 10 centimeters could be more than enough. So this AP release in response to heat, alarm pheromone release in response to heat, could also be happening in the field when PMPs are treating the apartments for bedbug control, right? And if some bed bugs in aggregation are kind of getting exposed to sublethal temperature, then they are going to release alarm pheromones that could signal the conspecifics that are in the vicinity to kind of escape that area. So we think we need to do more experiments in this, more practical experiments, but at least based on the lab results, we think the alarm pheromone release by heat exposed bed bugs could be driving their escape from lethally heated areas. Because we were able to get a similar response from synthetic AP blend, which is the hexanal and octanol, there is a potential that we could use alarm pheromones, synthetic alarm pheromones, to flush out bed bugs from their hiding spots, either before or after heat treatments, or also during insecticide treatments. So that research is probably Aaron is going to continue that research in his lab. That's definitely very interesting. So with that, I would like to conclude everything that I have presented today. Not necessarily conclude everything because some of the research is still ongoing, but just give you a summary of the different experiments that I presented about today. So first, at the population level, when we are looking at the physiological heat resistance development in bedbugs, we did not find any evidence that field collected bedbugs were developing physiological heat resistance. However, we did find that bedbugs were able to behaviorally respond to sublethal and lethal heat. And as soon as the temperatures of their harborage reached anywhere between 45 to 49 degrees Celsius, they were kind of randomly leaving their harborage in search for cooler areas. And about 4 to 12% of bedbugs were able to randomly find cooler areas, cooler spots, and then survive heat exposure. So this could also be happening in the field. And it is likely that even after doing everything correctly during a heat treatment, PMPs could see about 5 to 10% of the bedbugs survive. The bedbugs that are escaping to these cooler spots, their recovery from sublethal heat exposure could be aided by upregulation of the heat shock protein genes that we showed through qPCR experiments, as well as we found evidence that alarm pheromones, the hexanal and octanol that are released by bedbugs in response to heat, could be playing some role in this behavioral escape response of bedbugs when they are exposed to heat. So this was a very, in my opinion, it was a very successful PhD project for my student. I'm going to continue some of the research on some aspects of this thermal biology of bedbugs, and he's going to continue some research on his own. So with that, that's all I had to present today. I would like to thank the Urban Entomology Group here at Purdue University. The top picture is that of the group from 2018. And now in 2021, that group has almost completely changed. I would like to thank some of the funding agencies, Housing and Urban Development, as well as Pest Management Foundation, as well as a big shout out to Dr. Aaron Ashbrook for doing, for bearing with me and doing all the experiments that we planned out and getting 3 papers out of it. So with that, I would like to take any questions that you may have. Thank you.
Thank you.
Thank you. I'll leave the slides on. That way, if I need to go back to some of the slides to answer the questions. Thank you.
Kang Lu, I'm going to unmute everybody.
Okay,
if
go
you
ahead.
can see.
I can probably see some of the comments as well.
Okay.
Yeah.
I have a question about the heat experiment.
Mm-hmm.
You mentioned that at the 45 degrees, the time to reach 100% mortality is about 23 minutes.
Yes.
Is this the time you start to put the vial into the water bath, or the temperature that reaching 45 degrees inside the vial?
No, it's a heat shock experiment. So the water is already heated to 45 degrees Celsius. So as soon as we are immersing the test tubes in the water, they could be reaching that 45 degrees Celsius temperature in maybe 5 to 10 minutes.
Yes, I know. But
Yeah.
what I mean is that the 45-degree temperature is the water bath, but the inside the well, it takes some time to reach 45 degrees.
Yes, yes. So
So-
it is combined. Yeah, we are not calculating the temperature in the
Okay.
test tube. Yeah. Yeah. So are there any questions in the comments? So one of the data that I did not present to answer Dr. Wang's question is we did an experiment where we were kind of exposing them to the room temperature water, and eventually it was kind of raised, the temperature was raised to 45 degrees Celsius for the water bath. So that was even a slower heat exposure experiment. And even then we did not see any difference in survivorship.
Right. So the temperature, sorry, I just want to continue that topic. So the bed baths you used were males, recently fed, or the combination of different stages?
They were males fed 2 or 3 days before heat exposure, I believe. Yeah.
Oh, okay.
So I think I can answer a question that came through comments. Someone has asked, what was your undergraduate focus in entomology? So my undergraduate was actually in agricultural sciences, and agricultural entomology was one of the subject during my undergraduate major. So I did not have any research focus, but it was agricultural sciences.
I have a question about the population that was like heat treated, like for resistance. Do we know what the mechanism is that caused them to become like unviable to survive because of their resistance to heat?
Yeah, so we don't directly know the mechanisms, but I think people have shown that as you expose bedbugs to heat, sublethal heat, they lose their symbionts, which is Wolbachia, and they are shown to supply vitamins to bedbugs. And that loss of vitamin supply can lower their fecundity. We could also see some developmental effects. So maybe some of the growth related genes, the chitin synthase, or the juvenile hormone kind of related pathways were affected, as well as their feeding was affected. They were not feeding to repletion when we were offering them a blood meal. Does that answer your question?
Yeah, it does. Thank you.
Mm-hmm.
So if anybody else has a question, you should be able to unmute yourself and ask.
There was one question that came through in the chat. I have that chat window open.
Okay.
What caused my work on caused you to work on urban entomology after having agricultural focus? So my interest was insect toxicology within the field of entomology. That's why I kind of started working and the project that I worked for my PhD happened to be with urban pests. So that's how I kind of made my way from entomology, insect toxicology to urban entomology. It was just because I worked with the urban pests for my PhD research.
So there is a lot of needs in urban entomology. So welcome students start working in urban entomology, right?
Yeah.
Hello, Doctor.
Yes.
Can you hear me well?
Yes.
Yeah. This is Ginger. I'm Dr. Wang's PhD student, and we met in NCUE.
Yes.
Yeah. I have one question about the heat selection. About the selection, you use the population, and I want to ask how you do the selection, and you only select the male or female or whole population, and do you like cross mating to the unselected population, for example, like susceptible strain or lab strain, something like that?
Yeah, so ideally, if you were to go back and repeat the study, we would use kind of a field collected strain because it has more genetic diversity as compared to a laboratory population that we use for selection. And when you are doing selection experiments, you have to use kind of mixed sex, so both males and females, because you want the survivors, you are only exposing them to sublethal temperature, exposure time combination. So you do want some survivors and you want those survivors to breed into the next generation. Then once they breed into the next generation, you can again expose them to sublethal temperature and time combination. So yeah, does that answer your question? And we did not do any
Yes.
crosses. Yeah.
Oh, no crosses.
Yeah.
Okay, because my question is like maybe in the field, some insect, I mean, bedbugs, they already have high tolerance to the heat, and they may be like close mate to some other, some haven't have any resistance. So yeah.
No, that's a
yeah.
very good question. So yeah, in the field, the surviving bed bugs that are maybe physiologically challenged somewhat because of heat exposure could be then mating with bed bugs that are newly introduced. And then the new infestation that they start is kind of very healthy, not impacted at all. So very good question.
Thank you. It's a good talk.
Mhm. Thank you.
So we have two more questions in the chat.
Okay. I can see them. Are there concerns about histamines or other chemicals becoming aerosolized by the heat treatment? So one, I don't know too much about histamines. Definitely the dust can become not aerosolized, but people can get exposed to the dust that contains bedbug histamines. But actually what the research that is being published on this topic actually shows that the heat treatments actually reduce the level of histamine in infested homes. So I don't think there is a chance of them histamines getting aerosolized by heat treatments. I may be wrong, but that's my understanding. I'll read another question that came through. When this project is completed, what would you like to do with your results in relation to future studies? So one of the thing that we definitely want to we want to put forward some recommendations for PMPs. So how can they minimize bedbugs from surviving or escaping heat exposure, right? So some of the research will be published as trade journal articles for PMPs, where we can give some recommendation to put traps next to the infested furniture. So that kind of practical research has never been done. So what I want to look at is during a heat treatment, if you place climb up traps, which were developed by Dr. Changlu Wang at Purdue, if you place those traps, can you see some bed bugs kind of trapped in those? So that would be a good indicator, as well as alarm pheromones show some promise that are released by bed bugs in kind of developing, combining them with some other techniques that we are using for bedbug control. For example, there was a paper from 2009, I believe, that showed that insecticide dust, the efficacy of insecticide dust was improved by when they were blended with bedbug alarm pheromones. So there is some opportunity there. One more question. Are there any techniques or skills you wish you learned or explored more in undergrad before entering into your field? I think I did my bachelor's and master's in agriculture sciences and entomology in India. So definitely there were not many research opportunities there. I don't want to kind of single out any particular technique, but if you can gain research experience in different areas of entomology during your undergrad, that was going to serve you better throughout your career.
Okay, we've got time for one more question if there is. And so I'm not seeing or hearing anything. So those of you that are not involved with the seminar class, I'm going to ask you at this point to log off and we will continue our discussion with the speaker, with the students. And let me do
Thank you. Yeah.
Yeah,